Today I'm going to talk about drug repurposing, why and how is it important, and then a definition. So firstly, it's called, as Angela quite rightly said, it's using approved drugs for a new purpose. So these are safe drugs, these are approved drugs, drugs that have been used potentially with a new indication in mind. So I'm going to answer the question, why use drug repurposing, talk about my research goal, which is looking at childhood leukemias, talk about traditional drug discovery versus drug repurposing. So our previous speaker mentioned some targeted therapies, so talk about how that's slightly different. Then answer this very simple question, does drug repurposing actually work? Then I'm going to focus on my program of research, which has been funded recently by CCLG and Little Princess Trust, developing repurposed drugs as new treatments for childhood leukemia. I'm going to talk about a drug screen that I did, quite a large drug screen of about 800 drugs, the identification of some novel drug candidates, and then focusing on one hit, just as an example of one that I've taken forward, and some encouraging data in childhood leukemia cells. So 4,000 children are diagnosed with cancer every year in the UK, which is more than 10 each day, quite a stark fact. And most commonly diagnosed cancer within children is leukemias at 31%. And although my work today is going to be focusing on leukemia, I just want to make the point that drug repurposing is a highly relevant approach for many different types of childhood cancers, and no different types of childhood cancers are sort of involved at this meeting as well. So it's definitely relevant to other ones, but for the purposes of this talk, I'm going to be focusing on leukemia. So childhood leukemia is a cancer of the white blood cells. And normally white blood cells, as we probably know, grow and repair in a really controlled way. So it's a very controlled manner. In leukemia, for example, though, the cells divide, but they don't actually mature properly. And what you find is an accumulation of immature cells, which aren't really doing their job properly. So you can see the bone marrow here, which is inside like long bones, such as your leg bones. Normal bone marrow, you can see the white blood cells here. In purple, they've matured properly, they're doing their job, they're fighting infection. And you've got lots of red blood cells around here, which are really important for carrying oxygen, and platelets, which are important for wound healing. Then you have a bone marrow here with an acute leukemia, and you can see there's a lot of white blood cells. However, they haven't matured properly, they're not doing their job properly, and a lot less red blood cells and a lot less platelets. So children with acute leukemias may present with things like bruising because the platelets aren't doing their job properly. And then obviously issues with red blood cells, such as maybe breathlessness or things like that. So there's two main types of childhood leukemia: acute lymphoblastic leukemia, or ALL, represented here in blue, and acute myeloid leukemia, or AML. So while ALL is a more common leukemia, definitely, 80% of cases, the outcomes for AML are much worse, which is about 15% of cases. So my work focuses on acute myeloid leukemia. So it's unlikely, and lots of experts agree, that it's unlikely that improvements in long-term survival will be achieved just by more intensive chemotherapies. And many people in this room also know the current treatments can produce severe side effects. So the program of my work is really focused on two areas: trying to identify new drugs for childhood leukemia that are effective against the leukemia, obviously, but also spur the normal cells and provide less side effects and a lot less toxicity to healthy cells, which is obviously another important area. So the drugs that are currently used to treat frontline to treat childhood AML are very old. They've been around for a long time. Cytarabine was first synthesized in 1959. It took about 10 years for it to be approved by the FDA, so again, a very old drug. Daunorubicin then was identified in the 1950s, and interestingly, it was isolated from soil-based microbes near a castle in Italy back in the 13th century. Idarubicin is a more recent drug. It's more potent than daunorubicin, but it's still over 30 years old. So new drugs are really needed whenever we think about the outcomes and the survival, and also the side effects as well, and these are very old drugs. So to develop a new drug potentially for childhood leukemia, there's generally two ways we can go down this. We can do a traditional drug discovery approach from scratch, or a targeted therapy. So basically what you do is, this is a cancer cell here in blue. So you're looking at a specific cancer alteration, for example, a change in the DNA. So what you're really doing is you're developing a new drug A, which is a specific drug against a specific alteration in the cancer cell. Drug repurposing is slightly different. You can look at a specific alteration, but instead of developing a specific drug A, what you do is you take a library of drugs which are already approved. These are much safer drugs that have already been approved, as I said, and you screen those against that alteration. Or alternatively, some data I'm going to present today, what you do is you maybe screen on the basis of an anti-cancer effect. So you can take childhood leukemia cells and some normal cells, for example, and screen these approved drugs against that. So although my work's very focused on repurposing, just to say that both approaches really do have their advantages and successes. So targeted therapies have really revolutionized cancer care, as we know. So it's not to say that this is better than this, it's just another method. So this is the pipeline for sort of traditional drug discovery. You can see it's a very long process. It can last up to about 15 years. It begins with laboratory studies, then preclinical studies, which can last up to about 5 years. Then we have medicinal chemical optimization, basically getting the drug ready for humans, which can take a long time. Admit studies, which is a short word, but can take a lot of work and a lot of painstaking work. This is basically looking to see how the drug is absorbed, how it's distributed throughout the body, how it's metabolized, eliminated, and for toxicology as well and side effects. If you get past this part, you then move into formulation and manufacturing, basically getting the drug ready for clinical trials, and then obviously we know a lot about clinical trials and finally approval. So it's a long drawn out process. Interestingly, oncology drugs or cancer drugs have the lowest likelihood of approval in clinical trials. So this approach is definitely attractive and a lot more interesting within in the cancer field. Because it's a long process as well, the average cost to develop a new drug from start to finish is around $2.5 billion, so it's a huge amount of money. Drug repurposing is slightly different because you still have to identify your novel activity in laboratory studies, but those are generally shorter because you have an idea of maybe how the drug is working in its original indication. You do your preclinical studies, but generally speaking then, because the drug's approved, it's been through its toxicity profiles and so on, we can then move much quicker into clinical trials. So it's a much faster process, and because it's faster, it's also much cheaper as well. So does it actually work? And the answer is yes, it does. There's many examples of how it's worked. This is one example from the pediatric population. It's called infantile hemangiomas. They're a common soft tissue non-cancer tumor found in children, about 3 to 10% of infants. So the traditional treatment, which is systemic glucocorticoids, is two issues. Number one, the effectiveness is kind of variable, and number two, there were some safety concerns as well. And in 2008, there's lots of studies kind of showed really exciting data with this drug called propranolol, which many would be familiar with, which is to treat heart conditions such as angina and blood pressure. And this kind of work really gave the impetus for a New England Journal of Medicine, which is a very prestigious journal, this study, which was published in 2015. And what this group showed was that 88% of patients showed improvement on the propranolol compared to the previous drug. And it's now the frontline treatment, which is amazing. This is an image of a 4-month-old boy with the condition. You can see the hemangioma here on the ear and the regression after 4 months of propranolol. There's many other examples as well, which you may be more familiar with. For example, aspirin's been repurposed initially from inflammation and pain now as an antiplatelet drug. Bromocriptine, which was often used to treat Parkinson's disease, is now being repurposed to treat diabetes. And interestingly, in our lab, we have some data to show bromocriptine has anti-leukemia effects as well. Methotrexate, which is often used to treat breast cancer, has now been repurposed to treat psoriasis and arthritis. And propranolol actually as well has some repurposing effect as a preventative in migraines as well. So just to summarize this part of my talk, why is drug repurposing important? Well, traditional drug discovery science has limitations. It is very exciting, but it does have limitations. Many drugs fail toxicity trials, and it takes a long time to develop, and it's high cost as well. Drug repurposing has decreased development time and costs, and the drugs have already gained approval by the government. And also another interesting point is that approved drugs have a wealth of retrospective data, so we can look back in the past and look at safety issues and things like epidemiological analysis as well, which is going to be important, one of the examples I show in a moment. How is it important? Well, repurposed drug libraries can then be used to screen childhood leukemia cells, or childhood cancer cells and models with maybe specific alterations. And another thing that our lab does as well is a thing called connectivity mapping. So we can do computer-based analysis as well, which can predict approved drugs then that might be effective against a childhood cancer, and then we can test that in the lab. So I'm going to talk about an example then, which has been funded by CCLG and Little Princess Trust that we're currently working on, which is repurposing drugs in this childhood AML. So just some workflow of the experiment. So basically what we did was we developed cell line models in our laboratory of a childhood AML. I can go into detail for that if anyone's interested, and adult AML as well for means of a comparison. I then used the library, which is a large collection of approved drugs, 760 drugs in total from this commercial library. I tested the drugs then at 3 different concentrations, which are quite low concentrations, 10 nanomolar, 100 nanomolar, 1 micromolar, at 2 time points, and I performed this screen using this robot, which is called Integra ViaFlow 384. So all the drugs came in a large 384 well format, and we were able to use a computer programmer to pre-program this robot, so a lot of the work was done independently to prevent any errors. This is the data. So basically what this donut plot shows is every drug in a condition is basically a dot. This green circle, or yellow circle around the inside, represents 100% viability. These are drugs that have had no effect at all on the childhood leukemia cells. So that's encouraging to us because we expected, obviously you don't expect lots of repurposed drugs to have an effect. The vast majority had no effect at all. This red circle around the outside then represents 50% viability, or drugs that have killed the childhood leukemia cells by half or more. So as you move outside the circle, the further you get from the center, the more effective the drugs are against the childhood leukemia. And the exciting thing for us is that we got 38 hits of 50% viability. So that's 38 drugs that were effective against the childhood leukemia. So obviously the question is, what are the drugs? So really encouraging to us. I've grouped them together in what they do. The first 12 drugs are standard of care drugs that are used to treat blood cancers today. So that was really encouraging to us that out of a screen completely blinded, we were able to pick, or we were able to sort of fish out 12 drugs that are used in blood cancers today. The three main drugs that I talked about that are used standard of care came out as hits: cytarabine here, daunorubicin and idarubicin, and there's other drugs such used for example, childhood ALL that came out as hits. So that was really encouraging to us that the validity of the screen was sort of confirmed because we were picking out these drugs. The next 12 then, in no particular order, are drugs that are used to treat solid cancers. So you've got fluoxuridine, which is used to treat colorectal cancer, methotrexate, which you may know, oxaliplatin, paclitaxel, and so on. So again, that kind of made sense to us as well, that we're picking out drugs that are used to treat solid tumours. But the most exciting thing was the completely novel drugs that no one had really been worked on before in cancer at all, never mind childhood leukemia. And these are the drugs. So the interesting thing again to us was that more than one drug came out in each group, if that makes sense, for a certain condition. So we had two drugs which were used to treat parasitic worms, albendazole and mebendazole, which are effective against the childhood leukemia cells, two antimalarial drugs, two drugs which were used to treat pre-transplant as immunosuppressants, two statins, and other drugs as well to treat certain other conditions. So the question was then, we had 14 drugs that were novel hits, and we have other people in our lab working on some of these hits, but the question was then for us, well, how do we decide which drugs to take forward? And what I did was a Matchett screen. So this is basically like a matrix, which is rich in nutrients that the leukemia cells grow in quite well. So what I did was I took the childhood leukemia cells, the adult leukemia cells, and normal bone marrow, which is totally normal cells. And basically what I did then is, so basically how this works is that live or living leukemias form these colonies or clusters of cells. So the more clusters of cells you have, the more kind of growth or viable the leukemia is. So just for a means of control, I added cytarabine, which is a frontline treatment in childhood AML. And you can see it was reasonably effective against the childhood AML and the adult AML, as you'd kind of expect, but there were still some colonies remaining. But it did show toxicity in normal bone marrow, and that makes sense again to us because we do see side effects with cytarabine in that population. So one of the drugs that we're currently working on is called mebendazole, and this is some of the data from it. And you can see it is as effective, if not more effective, than the cytarabine in the childhood leukemia cells. It was very effective against the adult, and really importantly to us, there was no toxicity in the normal bone marrow. So if you remember from what my goals are for our research is to try and find drugs that are effective against the childhood leukemia while sparing the normal cells. This is obviously preliminary data, but it's very encouraging to us. We've taken this work forward. We've used, I'm not going to present the data today, but we've used many different types of childhood AML cell lines taken from 1 and 2 year old children in the laboratory, and we've tested those, and again shown very low concentrations are effective. These are very low concentrations, which I'll talk about in a moment. We've also taken normal cells from adult patients who are totally healthy, and we've shown no toxicity in those cells at all. So a lot more work to do, but it's very encouraging so far. So what are mebendazole and albendazole, which are two of the drugs that were taken forward for further analysis? So again, something that's interesting is that these drugs are already used within the pediatric population, most predominantly. So they're antihelminthic drugs. Basically what they're used to do is to treat children with parasitic worm infections. When children come with parasitic worm infections, they're given this drug one-off, and obviously that might not be suitable for leukemia treatment, but there is certain conditions that where children are treated long-term with this condition, and this important study showed that an oral drug, only 3 of 17 patients had any minor reactions, and those were things like skin conditions. So it's a very safe drug. Through personal communications with local GPs, they've been prescribing these drugs to children for 20, 30 years, and they have very, very little side effects, and we plan to go back and mine that data as well in our research. Other groups are kind of finding out as well that there is some activity with this. So people are starting to report anti-cancer activity in colorectal cancer, melanoma, and lung cancer. And for the scientists in the room as well, the drugs work by binding to this colchicine site of tubulin. So they work differently from other anti-childhood leukemia drugs that are currently there, but they also work differently from the likes of paclitaxel, which are microtubule inhibiting drugs. Also, again, encouraging to us is that the plasma concentrations from taking standard dosages in plasma are around 500 nanomolar for albendazole and 3.5 micromolar for albendazole. And what that basically means is that the concentrations that we see that are effective against the childhood leukemia in the laboratory are able to be achieved by standard dosages, but obviously there's more work needed to be done to see if we go intravenous or another route for that. This isn't a childhood cancer case, but this is an adult AML case of a relapsed AML. So this is someone who presented in Belfast with a relapsed AML, which basically means, as you know, that their leukemia unfortunately came back. And when it does come back, it's often more aggressive and resistant to treatment. And what we were able to do is get some cells from this patient, this male patient, and put them into this methylcellulose assay and treat them with albendazole. And again, at low concentrations which are achievable by the oral dosages, we saw efficacy in this as well. And we've taken this work forward. We're trying to work out now how the drug actually works, the mechanism as well, and doing some in vivo experiments and other preclinical work, because it's really important to figure out how the drug's working. So in conclusion, new therapies for childhood AML are required that will be effective against the cancer but spare normal cells, so equally both important. Traditional drug discovery is very, very exciting and has many successes, such as imatinib in CML. It's expensive, it's time consuming, and few drugs are approved. Drug repurposing is really, really gathering momentum, both within the scientific field, within the clinical field as well. It's really an alternative strategy. It's identifying new applications for existing or approved drugs, drugs that are licensed and mostly safe. What we did was we performed a large-scale screen of repurposed drugs in childhood AML cells. 38 drugs were identified as hits. 12 of these are standard of care drugs in blood cancers, including 3 which are standard of care in childhood AML, again, providing validity. 14 are totally novel. One of those drugs, we're working on many different drugs at the moment, but one of those drugs, albendazole, is effective against the childhood leukemia cells in the laboratory, many different types of childhood AML cells, while having little effect on normal cells, both from the patient and from the mouse as well. Our current work, as I've said, is focused on understanding how this drug works. So in conclusion, I want to thank our lab, and particularly Professor Kim Mills, who heads up our lab, Almac Discovery, who are also collaborating with this work, our colleagues in Dublin. Delighted that Wendy's here today. She's funded this work through Little Princess Trust and CCLG, Ashley and the team there. So we're really lucky to have Katrina Lappin, who's a really talented PhD student. She's been successful in getting the post that's funded by these guys, so she's going to be starting with us in September, and she's here today as well. Locally, I'm funded by Cash for Kids Northern Ireland and Leukemia Lymphoma Northern Ireland, who fund a lot of my work. So thank you very much.